Good morning to everyone. So today's class is to be trematodes. So coming to today's class, there are different types of helminths. So you all know about trematodes, nematodes and cystodes. So today we will be going to discuss about trematodes. So as cystodes means it is tapeworms, and trematodes those are flukes, and nematodes those are roundworms. So today I am going to focus about trematodes. So trematodes, the other names are flukes. And coming to the morphology and classification and history, so these are flat, fat and fleshy, and the appearance is leaf shaped worm. So these are called as flukes, and these are named as hermaphrodites, means monoecious, means all the male and female sex are in same fluke or in the same worm. There is no different male and female. So coming to the general characteristics of trematodes, these are leaf-like structure and it is broad or flat worms. So these are named as flukes. The size varies from 1 millimeter to several centimeters. The most general characteristics are external structures are two suckers. One is oral sucker where the intake is taken, and the other one is ventral suckers. And these are already I have mentioned monoecious. Both male and female sex are in the same worm, and the body is covered by a integument which is often called as bears the spines. And it doesn't have a body cavity, but still it has a circulatory system, respiratory systems like respiratory organs, and alimentary system which consists of mouth where the oral suckers are present, and a musculopharynx and the esophagus where the intake and everything goes into the gut, and anteriorly the ventral suckers are situated. And coming to the alimentary canal, it is appears as a inverted Y shaped, and absent of anus is seen. So coming to the excretory system, mainly it consists of flame cells and collecting ducts which leads to the bladder opening at the posterior end, and nervous system is also present and it has a paired ganglion cells. And coming to reproductive system, it is well developed in the flukes, and the genital organs lies between the two branches of the intestines. So coming to trematodes, all the trematodes are oviparous. Oviparous means which lays eggs. Only the trematodes, cystosomes are not a oviparous. Rest all the trematodes are oviparous. So coming to the trematodes, there are different types of trematodes. First one is cystosomes and facial hepatica, faciolipsis, busci and opistrosis or clonorosensis and paragonimus westermani. These are the scientific names. So coming to the common names, the blood flukes are called as cystosomes, and liver flukes are called as fasciola hepatica, and intestinal flukes are called as fasciola busci, and Chinese liver fluke is called as opistrosis or chlorinosis, and lung fluke paragonimus westermani. So today I am going to finish liver flukes. So the next coming classes we will study about the other intestinal blood flukes and lung flukes. So if you see the picture here, the fasciola hepatica is broad and it is a single organ, so it is broad. So these are called as flukes. Whereas in the worms we see round worms which is having unsegmented. In the cystodes where we can see the centimeters or tapes on the thing. So these are flat. So coming to the trematodes, the definite host is always humans, and all are oviparous which lays eggs. Coming to the intermediate host, we have two different types of intermediate host. First intermediate host and second intermediate host. The first intermediate host is cystosomes, and second intermediate host is other flukes. So coming to the first intermediate host, always it can be snails, and coming to the second intermediate host, it can be various like water plants in the fasciola hepatica, and in the fish it is for chlorionensis or opistроsis, and paragonimus westermani, the crabs are the second host. So coming to the classification of trematodes, there are different types of classification based on the organs where the trematodes is infected. So coming to the blood trematodes, mainly the blood trematodes are caused by Schistosoma hematobium, Schistosoma monsoi, Schistosoma japonicum. So all the schistosomes all comes under blood trematodes. So coming to Fasciola hepatica, opistrosis and chlorininescence, all are hepatic trematodes, so which causes liver infections. So today we are going to study about only the liver infections. So coming to the intestinal trematodes, mainly it is caused by again Fasciola, Buschke and Westermanni Watson. So coming to lung trematodes, only Paragonimus westermanni. So these are the trematodes and these are the habituants, or this is called as classification of trematodes. So coming to the first trematode, that is liver flukes. The other name, scientific name is hepatic trematodes. So mainly flukes are inhibiting the human biliary tract, which includes the entire liver hepatic system. So the organism which causes are called as Fasciola hepatica, Chlorinensis sinensis and Opistrosis sepsis. So all these species are going to be infected the lungs, liver. So these are called as liver flukes. So coming to the first organism, that is Fasciola hepatica. The common name for Fasciola hepatica is sheep liver fluke, or commonly called as even liver flukes. So coming to the adult worm, mainly it is present or it lives in the biliary passage of liver of definite host. So mainly here the definite hosts are sheep, cattle, goat and human beings, man. So morphology of the Fasciola hepatica, it is in two forms, adult worm and eggs. So coming to the adult worm, mainly it is broad, so leaf like structure, and it is called as flukes. It can be in the color of brown or grey in color. So mainly it is a bilateral symmetrical and mainly body cavity is present. So coming to the conical projections, mainly it is present in the anterior end and it is rounded at the posterior end, and the oral suckers are situated in the conical projections and the anterior end, whereas the ventral is present at posterior end and below the shoulders. The intestinal ceca and testes are extensively branched, and the lifespan of this adult worm is around 10 years. So this is how the adult worm looks like. It is flat and it appears like a leaf like structure, and the oral suckers are present in the anterior and the ventral suckers are present at posterior, and both it is monoecious, male and female are present in the same worm. So coming to the eggs is a large and oval, operculated, and it is a bile stained. So normally parasitology we study about bile stained and non-bile stained eggs. If the bile stained eggs are present, then it is yellow in color. So coming to the each egg contains a immature larva. Inside the egg there will be a larva. So that is the miracidium. So this is the infective stage of Fasciola hepatica. So eggs cannot be differentiated from Fasciola hepatica and Fasciola buschke. So this is the egg which is oval shape and it is a bile stained egg. So coming to the life cycle of Fasciola hepatica, we have one definite host, that is sheep, man, goat and man are the definite host where the sexually cycle takes place. Coming to the intermediate host, we have two types of intermediate host. First intermediate host is snails. That to the species is Lamniae is the species which is the name of the snail. And coming to the second intermediate host, mainly it is aquatic vegetative or water cercaris. So these eggs are passed out in the definite host, that is humans or sheep or cattle. So inside that feces there will be eggs present, and the eggs will be having the miracidium, so which is the infective form. So these feces are mainly mixed or enters into the fresh water either in the lakes or canals, and the embryo matures in the water in about 10 days and freely swims in the miracidium shield, and it is ciliated and passes through the stages of sporocytes. There will be first generation and second generation of radia stages which becomes a cercaria. Now this cercaria escapes and goes into the snails, that is first intermediate host, and then the water and enzymes of a aquatic vegetative becomes infective metacercasis, and these plants or metacercasis which is present in the aquatic, if it is consumed by the definite host like sheep, goat, man, so there the metacercasis enters into the duodenum, it is intestine, and passes into the gut wall and enters into peritoneal cavity. From there it penetrates into the capsule of the liver, ultimately it settles in the biliary tract or passage and matures into a adult worm, and it is going to produce again eggs within 3 to 4 months. So here you can see the better picture of the life cycle. So an embryonated egg which passes in the feces. So this is the egg which consists of miracidium. This is shedded with the definite host. It can be from man, human, sheep and goat. So this is mixed in the water, that is fresh water, lake or canals. There the snails is going to be consumed this egg, and the miracidium which is present inside this undergoes the development, and the miracidium is coming out from the snail which is a infective form. Again this is entering into the snail and it develops into a cercaris, and first stage and second stage of radia and sporocyst are formed. So this sporocyst then forms into a cercarious cyst which is present on the aquatic vegetables and the lake water. So now this is the infective stage. So once this miracidium is are infected with the aquatic cercaris, and when these water plants or water cercaris are mainly consumed by the humans, cattle, goats, and the adult worm is entered into the body of the gut wall or the liver of the definite host, and from there the adult worm takes 10 years to develop, and the eggs has been hatched from the definite host. This is how the life cycle repeats. So mainly Fasciola hepatica are mainly caused by aquatic vegetatives or water cercaris, and the stools are passed, eggs are passed in the stools, and the adult worm is like flat, so it is flukes, and mainly it is present in the liver, so it is named as Fasciola hepatica. So coming to the pathogenesis of Fasciola hepatica, the infection is called as fasciolasis, means infection of Fasciola hepatica, mainly which occurs in the rural areas where the sheep, goats have been herded, and mainly it injures the liver parenchyma and other biliary tracts. So mainly the symptoms of fever, abdominal pain, tenderness, hepatomegaly and jaundice are the common symptoms seen in the fasciolosis. And larvae migrants are mainly causes peritonitis cavities which is lodged in the ectopic foci, and formation of abscess are seen in the liver, and this site can be included even with the blood vessels, lung, subcutaneous tissues, brains and other orbits also. So coming to the laboratory diagnosis of Fasciola hepatica, we are going to demonstrate the eggs in the stool, or even we can aspirate the bile and we can demonstrate the eggs. So these eggs are bile strained eggs, and usually we can go for the blood test. It is a eosinophilia where we can see eosinophil count is increased, and we can go for serological tests also. ELISA test is a sensitivity test and it can be positive within 2 weeks of the infection. So coming to the treatment of Fasciola hepatica, orally we can take buthionol is the drug of choice or treatment of choice, and tricamizole and dihydromethane are the drug used for the treatment of fasciolosis. So coming to the prophylaxis, mainly health education is important for the sheep, cattle herders, and avoid of eating a raw or uncooked aquatic vegetables, and treatments of infected animals should be treated, and destruction of molluscum is important. We can use the molluscum sites which will kill the molluscums or snails. So coming to the other family or other species, Opistrosis sinuses, this also mainly causes liver infection or liver flukes. Mainly it is by using again eating undercooked fresh water fish. So mainly again these stools is also passed The eggs are also passed in the stool, and adult flukes are mainly present in the liver as well as gallbladder. Again, the adult worm is broad, same as the anterior end and posterior oral sucker, ventral sucker. This is also a monoecious. Both male and female are present in the same worm. So coming to Opistrosis senesis, some species of Opistrosis senesis can cause human infection, and Opistrosis flaninensis is also called as cat liver fluke, which is commonly seen in the Europes, and infection usually is asymptomatic and sometimes causes liver diseases which resembles Cloneriensis. So one more species of Opistrosis is vivarensis, which is commonly seen in the Thailand. So all these causes same infection like fasciola hepatic. So coming to other species, Fasciola gigantica. Fasciola gigantica has been reported from different countries like Africa, Asia, Russia, Iran and Hawaii. This life cycle is similar to that of Fasciola hepatica, but the intermediate hosts are different from snails. There the snail is involved, the name of the snail is Lisme. Here in the giganticum the snail is different, but the other diseases treatment life cycle is same of Fasciola hepatica. So coming to the other worm, that is Chlorionensis senesis. So this is again important in the liver flukes. The common name for this Chlorionensis senesis are Chinese liver fluke and Oriental liver fluke. It can be either named as Chinese liver fluke or Oriental liver fluke, and this parasite is endemic in Japan, China, Korea and Taiwan. Adult worm is mainly located in the biliary tract. So coming to the morphology of Chlorionensis, adult worm and egg. These are the two forms we see in the worm. And the adult worm, it is narrow, oblique, flat, and it is pointed at the anterior end and rounded at the posterior end. The oral suckers are mainly slightly larger than the ventral suckers. The blind intestinal ceca extends from the caudal region. It has a two large deeply lobulated testes which is present behind the posterior third of the body. So coming to the eggs, again these eggs are broadly ovoid and bile stained eggs, and it has a operculum at the one pole and small knob are present at the other end. Mainly it consists of ciliated embryo. Inside the egg we can see the miracidium. So this is the infective form in the Chlorionensis senesis. It doesn't float in the saturated salt solution. There are some concentrated methods, so saturated solution, so in that the egg will not be floated. So this is the egg, one end is narrow and the other one end is rounded, and it is a bile stained egg. So coming to the adult worm, the oral suckers are present at one end and the ventral suckers are at posterior end, and both the male and female are present in the same worm, so monoecious. So coming to the life cycle of Chlorionensis, definite host is man, pig, cat, dog and rat. Here also same intermediate hosts are two. First intermediate host is operculate snails and second intermediate host is freshwater fish, mainly lake fish and canal fish. The adult worm are first located in the biliary tract of the definite host. It can be present in man, pigs, cat or dog, rat, everything, and eggs can contain miracidium and discharges in the feces. So now these feces does not hatch in the water, but it is ingested by the snails. There are different types of snails, Bulimus, Paraphilorus, Seminiphilorus. All these snails are mainly involved in the Chlorionensis infection. Now this miracidium passes through the stages of first generation and second generation of Radia and Cercaria. So now this Cercaria escapes from the snail and enters into the water, that is fresh water or lake water or canals. So now this Cercaria which is present in the water will be intaken by the fish. So these fish, the Cercaria sheds their tails and insist under the scales in the fish. So this Metacercaria is a infective form of chlorionensis. So this is now present in the fish. While the definite host is consuming the fish, if it is a raw fish or raw undercooked fish, if it is not boiled properly, so the human beings or the definite host will be infected by these fishes and this Metacercaria. So this Metacercaria directly enters into the duodenum of the definite host. So coming to the life cycle, you can see in the picture the embryonated egg first passes in the feces of definite host. It can be either human beings, pigs, rats and so. So this embryonated egg will passes in the feces. So now this egg enters into the freshwater lake, that is, so inside this egg Miracidium is present. This is converted into sporocytes and first generation and second generation of radia is seen and Cercaria is totally seen. So now after this Cercaria is developed, so it enters or engulfed by the snails, and now the snails is again further Cercaria is enriched or comes out from the fresh water. From this fresh water, if the fishes are consuming this Metacercaria, so this is the infective form, and while we are consuming the fish, now the fish has a Metacercaria present inside the fish. So now we are consuming the undercooked fish or raw fish. So human being, that is definite host, is infected with the Metacercaria. So mainly this Metacercaria directly reaches to the duodenum and it causes chlorinosensis. And in the duodenum, inside the egg, the adult worm is formed. After adult worm is formed, again they start hatching the eggs, and in the eggs are passed in the stool. So first the human beings are the definite host, cat, rat, dog, all these are definite host. From definite host, the stool is passed. In the stool, the egg is passed. Inside the egg, we are having adult worm. Right. So in the worm, this enters into the fresh water. This is consumed by the snails and undergoes different stages, first and second generation of radiae, Cercaria and Metacercaria. Now this is Metacercaria is the infective stage. So this infective stage is mainly consumed by the fishes, and now inside the fish we are having Metacercaria. Now the definite host is consuming the fish and the definite host is infected by the Metacercaria. In the definite host, mainly the Metacercaria reaches into directly duodenum, and after few months the eggs will start converting into adult form, and again that adult form will hatches eggs, and that eggs is passed out in the feces. So coming to pathogenesis, mostly it causes again low grade or chronic type of minor symptoms like abdominal distress, intermediate diarrhea, so vomitings and motions can be seen, and liver pain and even tenderness. So coming to chlorionensis senescens, mainly it is linked with the bile duct carcinoma, even it can be causes carcinoma. So mainly it is a endemic diseases seen in Japan, Thailand, all these countries. So coming to the laboratory diagnosis, demonstration of eggs is mainly seen in the stool or even in the aspirated bile, because here it is livers are infected and biliary tract is also infected. Again eosinophilia, in differential count we can see the rise of eosinophils, that is eosinophilia. Coming to the serological test, antibodies can be demonstrated, either ELISA test or complement fixation test or indirect heme agglutination test. So coming to the skin test, intradermal test can be done using the antigen, that is extract of the chlorionensis senescens, so which is derived from the parasite, and it is kept as an antigen. It is injected intradermally and we see for the induration and erythema. So coming to the treatment, praziquantel has been reported to be the useful drug for the treating of chlorionensis. So coming to prophylaxis, mainly health education is very important in the rural areas. We have to educate the workers who are handling the cattle, pigs and rats, all these things, and avoid of eating raw or undercooked aquatic vegetative thing or water circaries, and proper disposal of feces is very important. If not the feces, the stool which is present, the eggs are present in the stool and mainly it dissolves or it enters into the fresh water, that is lakes and canals. So destruction of molluscum is very important. So we can use the molluscicides which kills the molluscum, that is snails. So this completes your lesson. Okay, so this is entire about liver flukes. So liver flukes are mainly Fasciola hepatica and opistrosis and chlorinensis. So these three are very important. In the examination view, Fasciola hepatica can be asked, even chlorinensis can be asked, but all the things are very important. So this completes your liver flukes. In the trematodes we have different types of flukes. So liver is completed. In later coming classes we will be studying about blood flukes and lung flukes. Thank you.